Welcome friends, welcome to the Knowledge Club Online. Now I am going to discuss about the messenger RNA is the carrier of information present in DNA. mRNA is transcribed in the nucleus. Processing it is transported to the cytoplasm and translated by the ribosome. Finally, the mRNA is degraded. Messenger RNA, mRNA, is a large family of RNA molecules that convey genetic information from DNA to the ribosome, where they specify the amino acid sequence of the protein products of gene expression. Following transcription of primary transcript mRNA, known as pre-mRNA, by RNA polymerase, processed mature mRNA is translated into a polymer of amino acids, a protein, as summarized in the central dogma of molecular biology. Genetic information is stored and transmitted in the nucleotide sequences of DNA. Selective expression of this information requires its transcription into mRNA that carries specific and precise messages from the nuclear data bank to the cytoplasmic sites of protein synthesis. In eukaryotes, the messengers mRNAs are usually synthesized as significantly larger precursor molecules that are processed prior to export from the nucleus. As in DNA, mRNA genetic information is in the sequence of nucleotides, which are arranged into codons consisting of 3 bases each. Each codon encodes for a specific amino acid except the stop codons, which terminate protein synthesis. This process of translation of codons into amino acids require 2 other types of RNA, transfer RNA, tRNA, that mediates recognition of the codon and provides the corresponding amino acid, and ribosomal RNA, rRNA, that is the central component of the ribosome's protein manufacturing machinery. Eukaryotic mRNA molecules often require extensive processing and transport, while prokaryotic molecules do not. Now come to the synthesis, processing and function of the mRNA. In transcription is when RNA is made from DNA. During transcription, RNA polymerase makes a copy of a gene from the DNA to mRNA as needed. This process is similar in eukaryotes and prokaryotes. One notable difference, however, is the eukaryotic RNA polymerase. Eukaryotic RNA polymerase associated with the mRNA processing enzymes during transcription, so that processing can proceed quickly after the start of transcription. The short-lived, unprocessed or partially processed product is termed precursor mRNA or pre-mRNA. Once completely processed, it is termed mature mRNA. Post-transcriptional modification. Processing of mRNA differs greatly among eukaryotes, bacteria and archaea. Non-eukaryotic mRNAs in essence mature upon transcription and requires no processing except in rare cases. Eukaryotic pre-mRNA, however, requires extensive processing. 5' cap. A 5' cap, also termed an mRNA cap, and RNA 7-methyl guanosine cap or an RNA M7G cap is a modified guanine nucleotide that has been added to the front or 5' end of the eukaryotic messenger RNA shortly after the start of transcription. The 5' cap consists of a terminal 6-methyl guanosine residue that is linked through a 5' triphosphate bond to the first transcribed nucleotide. Its presence is critical for recognition by the ribosome and protection from RNAs. Cap addition is coupled to transcription and occurs no transcriptionally, such that each influences the others. Shortly after the start of transcription, the 5' end of the mRNA being synthesized is bound by a cap synthesized complex associated with the RNA polymerase. This enzymatic complex catalyzes the chemical reactions that are required for mRNA capping. Synthesis proceeds as a multiple-step biochemical reaction. Splicing, genetic splicing is the process. Splicing is the process by which pre-mRNA is modified to remove certain stretches of non-coding sequences called introns. The stretches that remain pre-RNA is modified to remove certain stretches of non-coding sequences called introns. The stretches that remain include protein coding sequences and are called exons. To prevent confusion, use these mnemonic exons contain base pairs that will be expressed in the proteins, and introns are the intragenic regions. Sometimes pre-mRNA messages may be spliced in several different ways, allowing a single gene to encode multiple proteins. This process is called alternative splicing. Splicing is usually performed by an RNA protein complex called the spliceosome. But some RNA molecules are also capable of catalyzing their own splicing. Editing. In some instances, an mRNA will be edited, changing the nucleotide composition of the mRNA. An example in humans is the apolipoprotein B mRNA, which is edited in some tissues but not others. The editing creates an early stop codon, which upon translation produces a shorter protein. What is polyadenylation? Polyadenylation is the covalent linkage of a polyadenyl moiety to a messenger RNA molecule. In eukaryotic organisms, most messenger RNA mRNA molecules are polyadenylated at the 3' end. But recent studies have shown that short stretches of oligoiridination are also common. The poly tail and the protein bound to it aid in protecting mRNA from degradation by exonuclease. Polyadenylation is also important for transcription termination. Export of the mRNA from the nucleus and translation. mRNA can also be polyadenylated in prokaryotic organisms, where poly A tails act to facilitate rather than impede exonucleotic degradation. Polyadenylation site mutations also occur. The primary RNA transcript of a gene is cleaved at the poly A addition site, and 100 to 200 A's are added to the 3' end of the RNA. If this is altered, an abnormally long and unstable mRNA construct will be formed. Transport. Another difference between eukaryotes and prokaryotes is mRNA transport, because eukaryotic transcription and translation is compartmentally separated. Eukaryotic mRNA must be exported from nucleus to the cytoplasm, a process that may be regulated by different signaling pathways. Mature mRNAs are recognized by their process modification and then exported through the nuclear pore by binding to the cap binding proteins CBP20 and CBP80, as well as the transcription and export complex TREX. Multiple mRNA export pathways have been identified in eukaryotes. In neurons, mRNA must be transported from the soma to the dendrites, where local translation occurs in response to external stimuli such as beta-actin mRNA. Upon export from the nucleus, the mRNA associated with ZBP1 and the 40S subunit. The complex is bound by a motor protein and is transported to the target location, neurate extension along the cytoskeleton. Eventually, ZBP1 is phosphorylated by SRC in order for translation to be initiated. Many messages are marked with the so-called zip codes, which target their transport to a specific location. In translation, because prokaryotic mRNA does not need to be processed or transported, translation by the ribosome can begin immediately after the end of transcription. Therefore, it can be said that prokaryotic translation is coupled to transcription and occurs co-transcriptionally. Eukaryotic mRNA that has been processed and transported to the cytoplasm, that is mature mRNA, can then be translated by the ribosome. Translation may occur at a ribosome's free floating in the cytoplasm or directed to the endoplasmic reticulum by the signal recognition particle. Therefore, unlike in prokaryotes, eukaryotic translation is not directly coupled to transcription. Now come to the structure of mRNA. The structure of a mature eukaryotic mRNA, a fully processed mRNA, includes a 5' cap, 5' UTR coding region, 3' UTR and poly A tail. First I come to the 5' cap. What is 5' cap? The 5' cap is a modified guanine nucleotide added to the front 5' end of the pre-mRNA using a 5' triphosphate linkage. This modification is critical for recognition and proper attachment of mRNA to the ribosome as well as protection from 5' exonuclease. It may also be important for other essential processes such as splicing and transport. Now I come to the coding region. Coding regions are composed of codons which are decoded and translated in eukaryotes using into one and in prokaryotes usually into several into proteins by the ribosome. Coding region begins with the start codon and ends with the stop codon. In general, the start codon is an AUG triplet and stop codon is a UAA, UAG or UGA. The coding regions tend to be stabilized by internal base pairs. This impedes degradation. In addition to being protein coding, portions of coding regions may serve as regulatory sequences in the pre-mRNA as exotic splicing enhancer or exonic splicing silencer. What is the untranslated region? Untranslated regions, UTRs, are section of the mRNA before the start codon and the stop codon that are not translated. Termed the 5' untranslated region, 5' UTR and 3' untranslated region, 3' UTR respectively. These regions are transcribed with the coding region and thus are exotic as they are present in the mature mRNA. Several roles in genetic expression have been attributed to the untranslated regions, including mRNA stability, mRNA localization and translational efficiency. The ability of a UTR to perform these functions depends on the sequence of the UTR and can differ between mRNAs. The stability of mRNAs may be controlled by the 5' UTR and or 3' UTR due to varying affinity for RNA degrading enzymes called ribonucleases and for ancillary proteins that can promote or inhibit RNA degradation. Translational efficiency, including sometimes the complete inhibition of translation, can be controlled by UTRs. Proteins that bind to either the 3' or 5' UTR may affect translation by influencing the ribosome's ability to bind to the mRNA. MicroRNAs bound to the 3' UTR also may affect translational efficiency or mRNA stability. Cytoplasmic localization of mRNA is thought to be a function of the 3' UTR. Proteins that are needed in a particular region of the cell can also be translated there. In such a case, the 3' UTR may contain sequences that allow the transcript to be localized to this region for translation. Some of the elements contained in translational regions form a characteristic secondary structure when transcribed into RNA. These structural mRNA elements are involved in regulating the mRNA. Some, such as the SECIS element, are targets for proteins to bind. One class of mRNA element, the riboswitches, directly bind small nucleus molecules, changing their fold to modify levels of transcription or translation. In this case, the mRNA regulates itself. Next, I come to the poly-A tail. The 3' poly-A tail as a long sequence of adenine nucleotide, often several hundreds, added to the 3' end of the pre-mRNA. This tail promotes export from the nucleus and translation, and protects the mRNA from degradation. Next, come to the monocystronic versus polycystronic mRNA. What is cistron? A cistron is a gene. The term cistron is used to emphasize that genes exhibit a specific behavior in a cis-trans test. Distinct positions or loci within a genome are cistronic. An mRNA molecule is said to be monocystronic when it contains the genetic information to translate only a single protein chain polypeptide. This is the case for most of the eukaryotic mRNAs. On the other hand, polycystronic mRNA carries several open reading frames, ORFs, each of which is translated into a polypeptide. These polypeptides usually have a related function. They often are subunits composing a final complex proteins, and their coding sequence is grouped and regulated together in a regulatory region containing a promoter and a operator. Most of the mRNA found in bacteria and archaea is polycystronic. As in the human mitochondrial genome, dicistronic or bicistronic mRNA encodes only 2 proteins. Now, the mRNA circularization. In eukaryotes, mRNA molecules form circular structures due to an interaction between the EIF4E and poly-A binding protein, which both bind to EIF4G. Bind to EIF4G, forming an mRNA protein mRNA bridge. Circularization is thought to promote cycling of ribosomes on the mRNA, leading to time-efficient translation, and may also function to ensure only intact mRNA are translated. Partially degraded mRNA characteristically have no M7G cap or no poly-A tail. Other mechanisms for circularization exist, particularly in virus mRNA. Poliovirus mRNA uses a coverleaf section towards its 5' end to bind PCBP2, which binds poly-A binding protein, forming the familiar mRNA protein mRNA circle. Burley yellow dwarf virus has a binding between mRNA segments on its 5' end and 3' end, called kissing stem loops. Circularizing the mRNA without any proteins involved. RNA virus genomes, the strands of which are translated as mRNA, are also commonly circularized. During genome replication, the circularization acts to enhance genome replication speeds. Cycling a viral RNA-dependent RNA polymerase, much the same as the ribosome, is hypothesized to cycle. Now, the degradation of the mRNA. First, the prokaryotic mRNA degradation. In general, in prokaryotes, the lifetime of mRNA is much shorter than in eukaryotes. Prokaryotes degrade messages by using a combination of ribonucleases, including endonucleases, 3' exonucleases, and 5' exonucleases. In some instances, small RNA molecules sRNA tends to hundreds of nucleotides long can stimulate the degradation of specific mRNAs by base pairing with complementary sequences and facilitating ribonuclease cleavage by RNase 3. It was recently shown that bacteria also have a short of 5' cap consisting of a triphosphate on the 5' end. Removal of two of the phosphates leaves a 5' monophosphate, causing the message to be destroyed by the exonuclease RNase E, which degrades 5' to 3'. Now the eukaryotic mRNA turnover. Inside eukaryotic cells, there is the balance between the process of translation and mRNA decay. Messages that are being effectively translated are bound by ribosomes, the eukaryotic initiation factors EIF4E and EIF4G and poly-A binding protein. EIF4E and EIF4G block the decapping enzyme DCP2 and poly-A binding protein, blocks the exosome complex, protecting the ends of the message. The balance between translation and decay is reflected in the size and abundance of cytoplasmic structure known as P-bodies. The poly-A tail of the mRNA is short-ended by specialized exonucleases that are targeted to specific messenger RNAs by a combining of cis regulatory sequences on the RNA and transacting RNA binding proteins. Poly-A tail removal is thought to disrupt the circular structure of the message and destabilize the cap binding complex. The message is then subject to degradation by either the exosome complex or the decapping complex. In this way, translationally inactive messages can be destroyed quickly, while active messages remain intact. The mechanism by which a translation stops and the message is handled off to decay complexes is not understood in detail. In this picture, two nonsense mediated decay in metazones, premature translation termination results in an unusual long 3' untranslated region UTR, often in conjunction with an exon junction downstream of the PTC. The assembly of the PTC surveillance proteins of frameshift suppressor 1, UPF1, UPF2 and UPF3 at the site of translation termination is guarded by the presence thereof eukaryotic release factor 1, ERF1 and ERF3, which mediate translation termination at a UAA, UAG and UGA codons, and can be enhanced by the interaction of a UPF3 with an exon junction complex, which is a protein multimer deposit on exon junctions during splicing. Transported with mRNA to the cytoplasm and displaced by translating ribosomes only if bound in the coding regions. Phosphorylation of UPF1 by the SAR or THR kinase SMG1 triggers nonsense mediated decay through any of three pathways: deadenylation independent decapping by mRNA decapping enzyme subunit 2 DCP2 left, endonucleoside cleavage by SMG6 center, or poly-A tail removal by CCR4 not right. Deadenylation independent decapping or poly-A removal leads to degradation through the pathway. Endonucleoside cleavage leads to 5' exonucleotic degradation of the 3' fragments by 5' to 3' exonuclease 1 and degradation of the 5' fragment through the pathway is depicted. Now come to the AU-rich element decay. The presence of AU-rich element in some mammalian mRNAs tend to destabilize those transcripts through the action of cellular proteins that bind these sequences and stimulate poly-A tail removal. Loss of the poly-A tail is thought to promote mRNA degradation by facilitating attack by both the exosome complex and the decapping complex. Rapid mRNA degradation via AU-rich elements is a critical mechanism for preventing the overproduction of protein kinase such as tumor necrosis factor, TNF, and granulocyte macrophage colony stimulating factor, GM-CSF. AU-rich elements also regulate the biosynthesis of proto-oncogenic transcription factors like CJAN and CFOS. Now come to the nonsense-mediated decay. Eukaryotic messages are subject to surveillance by nonsense-mediated decay (NMD), which checks for the presence of premature stop codons, nonsense codons, in the message. These can arise via incomplete splicing, VDJ recombination in the adaptive immune system, mutations in DNA, transcription errors, leaking scanning by the ribosome causing a frame shift, and other causes. Detection of a premature stop codon triggers mRNA degradation by 5' decapping, 3' polytail removal, or endonucleoside cleavage. In this picture, only the 3' most exon-exon junction within a generic mammalian mRNA is shown. Mammalian mRNAs generally have an average of 7 to 8 splicing generated exon-exon junctions. A premature termination codon (PCT) that is located in the region initiated in blue, which is following by an exon-exon junction more than 50 to 55 nucleotides NT downstream. It is nonsense-mediated mRNA decay (NMD), whereas a PCT that is located in the region indicated in a green phase to elicit NMD. The normal termination codon TAR usually resides within the 3' most exons. In this picture, the nonsense-mediated mRNA decay is initiated into during a pioneer round of translation when a premature termination codon (PCT) resides more than 50 to 55 nucleotides upstream of an exon-exon junction that is bound to the exon junction complex EJC and EJC associated UPF2 and UPF3, UPF3X proteins as well as UPF1, which does not stably associated with mRNA, trigger NMD. The triggering mechanism remains to be resolved, but it presumably involves one or more component of the translation termination complex. Data indicate the NMD initiates from the mRNA 5' end, where it involves decapping followed by 5' to 3' exonucleolytic decay, and from the mRNA 3' end, where it involves deadenylation followed by 3' to 5' exonucleolytic decay. At present, it is not possible to determine the relative efficiencies of 5' to 3' and 3' to 5' decay during NMD. Next is a small interfering RNA, siRNA. In a metazone, small interfering RNA processed by dicer are incorporated into a complex known as the RNA-induced silencing complex, or RISC. This complex contains an endonuclease that cleavages perfectly complementary messages to which the siRNA binds. The resulting mRNA fragments are then destroyed by exonucleases. siRNA is commonly used in laboratories to block the function of genes in cell structure. It is thought to be part of the innate immune system, that is defense against double-stranded. In this figure, small interfering RNA induced degradation of mRNA in the cytoplasm comprises 4 principal steps. ATP-independent incorporation of siRNAs into the multi-subunit RNA-induced silencing complex. ATP dependent unwinding of the siRNA duplex, ATP independent binding to mRNA, and irreversible cleavage by RNA's 3' activity as the first step of mRNA degradation. Antisense oligonucleotides function as gene inhibitors by one or several mechanisms depending on the chemical composition of the oligonucleotide. The most commonly used examples are partially modified ASOs, which are delivered to cells in a single-stranded form and include RNase H to bind to the mRNA oligonucleotide complex in the cytoplasm and nucleus. RNase H cleaves the mRNA progressively. Both processes occur with catalytic amounts of oligonucleotides. Although under contents of cellular transfections, oligonucleotides are often present in excess quantities over the largest target mRNA. The principal difference between the two mechanisms is the nature and role in the enzymes. However, the interactions of both these these types of oligonucleotides with the mRNA follows the rules of Watson-Crick recognition. Next, I come to the microRNA, miRNA. MicroRNA are small RNAs that typically are partially complementary to sequences in a metazone messenger RNAs. Binding to a miRNA to a messenger can repress a translation of that message and accelerate poly A tail removal, therefore hastening mRNA degradation. The mechanism of action of miRNAs is the subject of active research. In this figure, the plant microRNAs that are bound to argonate recognize mRNA targets containing fully or nearly complementary binding sites, which are predominantly located in the ORF. The B figure by AGOs cleaves the mRNA in the base paired region between nucleotides 10 and 11 opposite the miRNA strand at the red arrow. The endonucleolytic activity of AGOs resides in the PIWI domain. The mRNA fragments resulting from the endonucleolytic cleavage are degraded from the newly generated 3' and 5' ends by the exosome and the exonucleus are xRNA4 respectively. xRNA4 is related to the major 5' to 3' exonucleus xRNA1 in animals. At the C picture, alternatively, the endonucleolytic cleavage by AGOs is prevented and the mRNA target is translationally repressed by an unknown mechanism, EIF eukaryotic translation initiation factor, PABPC cytoplasmic poly A binding protein. Other decay mechanisms, there are other ways by which messengers can be degraded, including non-stop decay and silencing by PI interacting RNA, piRNA, among others. mRNA-based therapeutics, various types of therapeutics are present. mRNA-based. mRNA is currently being investigated for its potential use in the treatment and prevention of diseases. mRNA-based vaccines are being developed as cancer immunotherapy and prophylactic vaccines for infectious diseases. mRNA is also being studied as a source of therapeutic gene products and protein replacement therapies in vivo, like cancer immunotherapy, infectious diseases vaccine, influenza, RSV, HIV-specific antigens, allergy tolerization, protein allergens, protein replacement and supplementation therapies, IL-10, EPO, SPB, VEGF-A, FOXP3, genomic engineering, transposase, ZNF, TLENS, etc. This lecture may be helpful to you. Thank you.